Hey folks, and welcome to another edition of Zog Science. Today we're going to start a little mini series on the central dogma, and we're going to begin with DNA replication. So basically, going off of our last unit, a whole bunch of people worked really hard to figure out what molecule stores genetic information. The answer is DNA. The next step was to figure out how DNA now takes that information, right, and then uses it for living things. How does it take that DNA and turn it into something that is, a living thing? And the answer is RNA and proteins. Now, how is that information stored? Well, information is stored in the specific sequences of the DNA nucleotides, or also known as bases, like a specific sequence of letters makes a word. Random sequences of DNA do not store successful information, right? Just like that random set of letters doesn't mean anything, right? It might store information, but doesn't store useful information. So DNA replication, there are three different possibilities for DNA replication. We can either have a conservative model, a semi-conservative model, or the dispersive model. In a conservative model, we are going to end up with one DNA molecule that will create a new DNA molecule, but that old DNA molecule will stay the same. It's not going to become a new one. It's not part of it's not going to go to the new one. In the semi-conservative model, we're going to end up with half of the DNA being made from one of the strands, the other half of the DNA being made from the other strand, and then it continues forward from there. We also have the dispersive model, which is basically that part of the DNA is going to always be kind of mixed around. So pieces of it are going to be on both strands of the new DNA. So Meselson and Stahl, they perform an experiment, and what they do is that they culture bacteria in a medium containing nitrogen 15 and some bacteria in a nitrogen 14. And what we're expecting is that we are going to see, if it's a conservative model, we're going to see two different bands all the time, because you're always going to have some DNA that is brand new and some that is old. In a semi-conservative model, we expect to see one band at the beginning, and then we see two different bands after one replication, the second replication. If it's a dispersive model, we should always have DNA being at the same spot. And the results of their experiment were that we ended up with one band at the beginning and two bands at the end, meaning that it is semi-conservative replication. So let's just kind of a little overview. The base pair rules are used to create identical strands of daughter DNA. So we've got the parent molecule separates out, and then the daughter DNA molecules are going to be added on, built off of the template so that they are identical to the parent and to each other. Now, question is, where does replication begin? Well, in prokaryotic DNA, which is circular, there is one place that is called the origin, and DNA replication begins at that point and then spreads out. So if you kind of look down here, right, you've got a circular piece of DNA. There is an origin, and from there, the new daughter DNA is made, and it proceeds forward in both directions. You can kind of see that here in this picture. So the origin was somewhere here in the middle, and it's replicating in both directions. We will eventually end up with two daughter DNA molecules. In eukaryotes, which is linear DNA, there are multiple origins along the way, and at each origin, a bubble forms and a replication fork occurs. So you have multiple places where DNA replication is occurring, but you still end up with two daughter DNA molecules. You can kind of see the two, the various replication forks everywhere that the arrows are kind of pointing, and this would be kind of the DNA that hasn't been replicated yet. So it's not just one place that replication is occurring. In eukaryotes, it's at many places. Now, the origin kind of details what we have is we have this a molecule called topoisomerase, which relieves pressure caused by the untwisting of the DNA. So DNA wants to be twisted. When we untwist it, there's some pressure, so the topoisomerase helps to kind of relieve that pressure. We have helicase, which untwists the DNA and breaks hydrogen bonds between the bases, so it unzips it. Then we've got some single-strand binding proteins. They're going to bind to the unpaired strands, keeping them apart and also making sure that they don't become unstable. Finally, we've got primase, which adds a complementary RNA primer to the template strand. So we can see that kind of being added here, and that allows DNA polymerase to then bind on. So it's a very important part of it. The next step is we have elongation. Elongation is where the DNA molecules are going to be added on, so we're adding on nucleotides. We can only add nucleotides to the 3' end of a DNA molecule, so we are building onto that end, and the nucleotides are added by DNA polymerase molecules. So we have two different strands. We have the leading strand and we have the lagging strand. And again, this is because we have this directionality of DNA, right? We can only add in the 5' to 3' direction. Therefore, we've got two different things that are going to be going on as the DNA unzips. Oops, too far. For the leading strand, the DNA polymerase 3 adds nucleotides continuously as the helicase unzips. So we get our RNA primer that gets added on. The DNA polymerase 3 kind of comes around it, and it begins to add on the DNA molecules using the RNA primer as kind of a starting point, and eventually it's going to look something like this, where it's continuing to add on, and it's just going to continue to elongate as the helicase unzips. It's just going to continue to add on because, you know, it's going in the proper direction. Now, the lagging strand, because DNA polymerase 3 can only add in the 3' end of DNA, one of the strands is slightly delayed. That's the one we call the lagging strand, and it's a little bit more complicated because of that. So what happens is that, again, we add the RNA primer. The RNA primer will then allow DNA polymerase to bind. DNA polymerase begins adding on nucleotides. The DNA polymerase then kind of reaches the next piece of RNA primer, and that is called an Okazaki fragment. It then pops off. A new RNA primer is added onto the other end. The DNA polymerase recognizes that, begins adding nucleotides until it reaches the next RNA primer, and it will then come back around. The next piece right here, this is going to be DNA ligase, DNA polymerase 1 will come in and replace that RNA primer, and then ligase will come in and it will bind the Okazaki fragments together. So what happens is it kind of does a piece and then a piece. As it is kind of being unzipped, it's just kind of adding on piece by piece by piece. So again, this is what that kind of looks like. We've got DNA polymerase 3, primase adding on primers, DNA polymerase 3 coming along, adding on nucleotides. DNA polymerase 1 comes in behind that, takes out the RNA primer. DNA ligase then comes in and adds all the Okazaki fragments together. Again, the leading strand just gets continuously replicated because as it's being unzipped, DNA polymerase 3 can just keep on adding on as it needs to. So I've got a little animation that will kind of explain it and kind of show it to you in a little bit different detail. Maybe. See if we can get it to play. There we go.
During DNA replication, both strands of the double helix act as templates for the formation of new DNA molecules. Copying occurs at a localized region called the replication fork, which is a Y-shaped structure where new DNA strands are synthesized by a multi-enzyme complex. Here, the DNA to be copied enters the complex from the left. One new strand is leaving at the top of frame, and the other new strand is leaving at the bottom. The first step in DNA replication is the separation of the two strands by an enzyme called helicase. This spins the incoming DNA to unravel it at 10,000 RPM in the case of bacterial systems. The separated strands are called 3' and 5', distinguished by the direction in which their component nucleotides join up. The 3' DNA strand, also known as the leading strand, is diverted to a DNA polymerase and is used as a continuous template for the synthesis of the first daughter DNA helix. The other half of the DNA double helix, known as the lagging strand, has the opposite orientation and consequently requires a more complicated copying mechanism. As it emerges from the helicase, the lagging strand is organized into sections called Okazaki fragments. These are then presented to a second DNA polymerase enzyme in the preferred 5' to 3' orientation. These sections are then effectively synthesized backwards. When the copying is complete, The finished section is released and the next loop is drawn back for replication. Intricate as this mechanism appears, numerous components have been deliberately left out to avoid complete confusion. The exposed strands of single DNA are covered by protective binding proteins, and in some systems, multiple Okazaki fragments may be present.
All right, now, one of the things that kind of to note is that how different this looks from the diagrams in the textbook, and that that's just because people don't really know what it actually looks like. They're just trying to come up with ideas for what they think it looks like based on research. So, just kind of something interesting to point out. So, now. Proofreading and repairing. Sometimes the nucleotides are mismatched, so sometimes they're not exactly put in there correctly. When the mismatch is identified, and there are some enzymes that do that, the offending DNA is cut out by a nuclease. The proper DNA is reattached by DNA polymerase, and then DNA ligase will come along and reattach things as it should be. Sometimes this process doesn't happen, and that is when a mutation occurs. But most of the time, I would say I think it's about 1 out of 100,000 bases is a mutation. So most of the time, these errors don't happen. Which, when you think about how quickly this is occurring and how much DNA is being replicated, it's really rather impressive. Now, at the end of the DNA, we have these ends called telomeres, and the telomeres are kind of a cushion at the end of the DNA. And the reason why is because on the lagging strand, we've got a little piece of RNA primer at the end that cannot be kind of replicated properly. And so what happens is that we end up with strands that are a little shorter, and we end up with shorter and shorter DNA molecules, right? So what we have is we have a molecule or an enzyme telomerase, which helps to replace that at the end. And again, telomeres have some links to cancer and aging that we kind of talked about in class. Cancer cells generally have overactive telomerase, so it keeps on adding on and acting on telomeres so that the DNA never actually gets shorter. Also, as your telomeres tend to shorten, that has been linked with aging, although the exact linkages haven't really been discovered quite yet. So we've got some information, right? We've got it stored. We're able to replicate that information. Now we need to take that information and change it into something useful, something meaningful, how organisms actually live. Enter the 2 T's, transcription and translation, which are all a part of the central dogma of biology. We've got DNA, which stores the information. It gets turned into RNA through the process of transcription, which is then translated into a protein, which codes for the traits. And we will be getting into transcription in the next video and translation after that. So thanks for watching, and I'll see you next time.